Okay, here's unit number 4, question number 2. This one is about cells leaving the G0 stage of mitosis and resuming participation in the cell cycle. And in this case, scientists are using a type of cell called a fibroblast to study how a cell might be induced, or how you can tell if a cell has been induced to leave G0 and enter back into the cell cycle. And to do this, they've made 7 experimental groups, 5 of which are in a 0.1% serum solution. One of those groups is in a 10% serum solution. One of those groups is in a 0% serum solution. And that's significant because we're given the information that in a laboratory, fibroblasts typically grow and divide when they are cultured in a growth medium that contains 10% serum, serum being the liquid part of the blood that remains after blood clots. Okay, if they go for 48 hours in the absence of serum, they leave the cell cycle and they enter G0. Okay, so to measure whether or not the cells are leaving G0 and entering the cell cycle, the scientists decide they're going to say they're going to measure what percentage of maximum DNA replication is the cell undergoing. And maximum DNA replication occurs here in group 2, which is bathed in a 10% serum solution. Okay, so the rest of these bars show what percentage of maximum DNA replication is happening in each of these experimental groups. Okay, and in addition, this chart is showing the presence or absence of 4 particular proteins, P16, 18, 21, and 27, which are, let's go ahead and read, 4 proteins that are inhibitors of cyclin-dependent kinases. Okay, so cyclin-dependent kinases, you may remember, become part of the CDK cyclin complex when that complex is created, it initiates the cell to move through the stages of mitosis. Okay, so these proteins inhibit cyclin-dependent kinases. So we would think that when they are present, they would stop the cell cycle from moving forward. Anyways, so those are some of the things that are being tested here. Let's look at part A, which says, based on the data, for the groups cultured with 0.1% serum, those are groups 3 through 7, identify which of the 4 proteins tested do or does not appear to play a role in blocking the cell cycle entry by fibroblasts. Okay, so positive means the protein is there, negative means it's not there. And I think it's pretty clear that protein 16 is not implicated in, how do they word that? Does not play a role in blocking cell cycle entry, meaning leaving G0 and entering the mitotic cycle, because these 4 experimental groups, 4, 5, 6, and 7, looks like they've all entered the mitotic cycle based on the amount of DNA replication that's occurring, and P16 is present in every one of them. That's really the only consistency between those. So that's the logic used for A. For B, it says based on the data for the groups of cells cultured with 0.1% serum, again groups 3 to 7, identify the groups most likely to begin undergoing a mitosis. I just kind of answered that. 4 through 7, in my opinion, you could make an argument that it's just 7. We don't really know what the criteria is for what percentage of maximum rate of DNA replication indicates that a cell is undergoing mitosis, but it seems like this cell in 0% serum is still replicating DNA at, let's say, 18% of maximum, but we know that that's not undergoing mitosis because of the information given up here. So we know that group 3 clearly is not. Okay, group 2 is not an option because it's not in 0.1% serum solution. So it would be either all 4 of these, or if you decide that only 7 is reached the threshold that indicates it's entered back into the cell cycle, fine, but one of those two options. Okay, C, neurons are in G0 and do not generally divide in response to an injury. Scientists predict they can use this data to help them treat spinal cord injuries, provide reasoning. So they're probably thinking that inhibiting P21 could help initiate the re-entry into the cell cycle, because that's a consistent marker here that gets cells to re-enter the cell cycle. Okay. Also, yeah, that's about it actually. That's all we got. Okay, and D, interactions between cyclins and CDKs control the cell cycle. Explain how the presence or absence of inhibitors might play a role in normal cells becoming cancer cells. And really what they're looking for is which of those options, the presence or absence of inhibitors, would allow cells to become cancerous. And in this case, the answer would be the absence of inhibitors would allow a cell to become cancerous, because an inhibitor stops the action of a CDK cyclin complex, which means if an inhibitor is present, it will stop the CDK cyclin complex from causing the cell to move through the cell cycle. Okay, but if that inhibitor is absent, it's sort of like taking the brakes away, right? There will be no brakes on, there will be no inhibition of the cell cycle, so it will go round, round, and round potentially, and end up dividing and dividing and dividing and dividing, and potentially becoming cancerous in that way. Okay.